All right, now let's talk about infectious disease. This is section 35.1, um, continuing our discussion on diversity. In this case, diversity of basically pathogenic organisms. Of course, there was a time when they didn't know about microbes. People would get sick. They'd have no clue why, really. But in basically beginning in the 1800s, people started to figure out that there were these tiny disease-causing organisms. Um, and these disease-causing organisms and the diseases they cause became known as infectious disease to differentiate from, say, something like cancer or a lot of cancers, which really aren't infectious. They can't be passed from person to person. So these are things that can move around and spread in a population, basically. Um, and these two folks, Louis Pasteur and Robert Koch, um, I have that spelled wrong. That should be Koch, not K. Um, they're the ones that came up, who did a lot of the early work on this and defined infectious disease when microbes cause physiological changes that disrupt normal body functions. And they developed what became known as the germ theory of disease. We'll put that in quotes because germ is not really a scientific word. It's sort of a vague term for something that causes disease, perhaps. Um, Now, you might recognize this, Pasteur, the process of pasteurization. This is when you heat up milk and other kinds of foods to kill pathogens. It was Louis Pasteur that figured out how to do this, basically, that you could purify or clean things by heating them up, um, or essentially sterilizing them. Now, so here are the infectious disease causing agents. Viruses, we've all heard of viruses, of course. Viruses come in various forms, but most basically they have what's called a capsid, which is a protein coat that essentially covers a nucleic acid, either RNA or DNA. And that's, at their simplest, that's what they consist of. The capsid, this protein coat, and either RNA or DNA inside. And there are RNA and there are DNA viruses. Now, some of them have an addition of having basically a covering over that, which is a membrane-like structure. And that's what you're seeing here with rabies and influenza and HIV, is that you have this membrane that covers it, and that membrane will have these proteins sticking out of it. Um, and that membrane and those kinds of viruses assist in entering host cells. Um, the goal of the virus, if you will, is to enter the host, infect cells in the host, get its RNA or DNA injected into those host cells, and to take over those host cells. For example, here's influenza, here's the virus. So inside is the capsid and in this case, oh no, hold on. All right, we got it plugged in. So here we have influenza virus. Again, inside is the capsid, and not sure. I think influenza is a DNA virus. And you have the membrane coat with these proteins sticking out, and together this all makes up this virus, or what they're calling a virus here. And it enters its host, and again, the goal is to get the nucleic acid out of the virus into the host, essentially um, to take over that host. And so the instructions in the viral DNA will be used to make essentially more viruses by that host cell. And then those new viruses that are made will be released or break out of that host cell, and then they'll be released from the organism and can spread and infect either other cells inside that host or outside the host, infect a different person. Um, Essentially, the cell again gets turned into uh, a biochemical machine that makes new virus, and it doesn't really do what it's normally supposed to do. Um, And when enough of those cells in a host get infected, that's when you start to have the symptoms and the problems of that infection. All right, so viruses. Now bacteria. So bacteria, like the infamous E. coli you've heard of, and Staphylococcus, these kinds of things, um, are another primary disease-causing organism. Here we have MRSA, methicillin-resistant Staphylococcus aureus. Methicillin is a type of antibiotic that these particular kinds of staph bacteria have become resistant, immune to. They've evolved over time, the resistance, and they've become a significant problem in hospitals and other settings. So many types of bacterial infections out there. Fungi can cause certain types of infections. Athlete's foot you've probably heard of. Um, histoplasmosis you probably have not heard of. While there are many, I should say, beneficial bacteria and fungi out there in the world doing things like decomposition and necessary ecological functions, there are some that cause disease, and so I want you to notice in this picture, this is sort of a microscope look at histoplasmosis. Fungi consist of these long strands and these structures they use to produce spores, and that's what you're seeing here. Um, all right, another disease-causing group of organisms, the Protista, as you're seeing with your pathogens project. Protista is one of the kingdoms we look at. They're generally unicellular organisms. They're not bacteria, they're not plants, animals, or fungi. They're sort of in between those groups. Um, and again, there are plenty of Protista out there that don't harm us at all, but there are some that do cause disease. Malaria is probably the most famous of these Protistan uh, pathogens. Amoebic dysentery, or Montezuma's revenge, which is this amoeba that basically lives in water, and polluted water in particular will tend to have more of it. And so if you happen to ingest it somehow, you can get extreme intestinal discomfort from that. Um, Guinea worm is, there are lots of these small worms that cause different infections. Guinea worm is a rather nasty one, particularly to Africa. Um, all right, so those are the disease-causing organisms again. Um, Again, viruses, bacteria, fungi, and protistas. Now, back to Koch, and here I've got it spelled correctly. These are the postulates that he developed to identify and determine that a microbe is indeed causing a disease. So first of all, you have to find that pathogen only in infected individuals, not healthy ones. You isolate that pathogen, you grow it in the laboratory, or you basically culture it. You then take that cultured pathogen and you introduce it into a healthy host. And if that pathogen is then isolated from that second host, which means it's basically living in them, you have basically determined that this organism indeed does cause this disease. Now, you might look at this one right here and say, gosh, why would you want to do that? Well, generally you'll do this kind of stuff on mice and rats and other mammals. You wouldn't necessarily do it on people. Um, all right, so Koch's postulates are essentially, again, used to determine that a particular microbe that you suspect causes a disease does indeed cause that disease. Now, there are other microbes that live inside of us that do no harm, and in some cases are beneficial. These are known as symbionts. Um, they have a symbiotic relationship, in particular a beneficial symbiotic relationship. And for example, when you look on the side of certain yogurts, these are all abbreviations for particular kinds of bacteria that are in that yogurt culture, but they're bacteria that are beneficial to us. In particular, in your gut, in your large intestine, you have a lot of microbes that live there and aid with digestion. They're sometimes known as your internal flora and fauna. Okay, all these organisms that live inside you. Now, one of them is E. coli. You have E. coli living inside of you. It's a relatively small proportion of all these microbes you have inside you, but they are there. However, you've probably heard of E. coli as a pathogen. Well, there are types of E. coli that, if you do ingest them, can cause basically food poisoning at a minimum, and at the worst, they can possibly kill you. Um, you get these bad E. coli, or essentially the bad E. coli, come from other animals. Mammals in particular all have E. coli living inside them. E. coli is the bacterium that lives inside of animals. If you have your kind of E. coli, that's good. But if you ingest E. coli from some other animal, that's when you can have problems. All right, we'll talk in a second how that can happen. How does diseases spread? Well, of course, through coughing and sneezing, and we all know we're supposed to cover our nose and mouth when we cough or sneeze so that you don't spread it, as you can see in this image here. It's kind of disgusting. Contact. So, for example, if you do sneeze in your hand, but then you shake somebody's hand, you can spread the microbe to them, and then if they were to stick their finger in their mouth, they can spread that way, which is, of course, why we wash our hands with some regularity, in particular before you eat. Wash your hands before you go to lunch. Um, now, exchange of body fluids. So, through sexual contact, STDs, sexually transmitted diseases. There are certain ones of these that don't really spread through just casual contact, but more intimate contact, you might say. So something like HIV, the human immunodeficiency virus that causes AIDS, um, certain types of herpes. These are diseases that, again, through casual contact are not spread, but through sexual contact. Intravenous drug use. When you use needles to inject drugs, and if you share those needles, and one person has a disease, it can spread to the other. Um, I'm having a moment right now. I'm trying to think of hepatitis. There we go. Hepatitis, particular hepatitis C, is a particularly nasty bloodborne disease that is spread through IV drug use. Um, as we were sort of alluding to with E. coli, when you have contaminated water and food, so if you're drinking water that has some of these things in it, that obviously can cause a disease. A well-known example of this is cholera, which is a bacterium that spreads in dirty water, particularly water that has animal waste in it. Um, and through contaminated food as well. So every once in a while we hear about food or food poisoning outbreaks, E. coli, other things from food that's been contaminated. I was just eating the other day I was going to eat some hummus, and my wife said, "Oh, there's been a hummus recall because there's this pathogen called Listeria that was found in some of them." Now, fortunately, we looked it up, and the brand and type we had was not part of the recall, but those things do happen every once in a while. I mean, when they process the food, sometimes the machinery has, it's not clean enough for whatever reason. Also, with some of these E. coli outbreaks, what can happen is the E. coli that comes from cows, of course, poop on the ground. That stuff can get washed into the water supply, say the river. If that water from the river is then being used to water fields of spinach or strawberries or something like that, then the E. coli can get on those vegetables. And then if someone eats those vegetables without having rinsed it off at all, or even if they did rinse it, sometimes it doesn't always do it, they eat that fruit or vegetable that has that E. coli in it, then it can make them ill. And of course, this is why you have to cook your meat thoroughly, particularly ground beef. You wouldn't really want to eat a rare hamburger because the heat, of course, kills these pathogens, but if you don't heat it up enough, it won't kill them. Um, Pathogens spread by certain animal vectors. These are known as zoonoses. It's just a pathogen that's spread by an animal is called a zoonosis. So malaria is a good example. It's spread by mosquitoes.